OrthoBunyaVirus. For the brief overview, in Uganda in Africa, there's a place called Bunyamira, and this is where these zoonotic viruses come from. They are arboviruses and they cause severe hemorrhagic disease. They are sensitive to detergents, but they have good antigens, and they cause hemorrhagic diseases, fetal abnormalities, and abortion. For the occurrence, the Simbo serogroup is the same disease caused by viruses with different geographical locations. The Epi1 virus is found in Asia, Australia, Turkey, Africa, and Israel. The Onyo virus is found in Japan and Australia. And the Schmallenberg virus is found in Western European countries such as Germany, Netherlands, Belgium, and the UK. So the OrthoBunyaVirus genus is a part of the Peribunyavirudae, which is a part of the bunyaviruses. These viruses are the Epi1 virus, the Onyo virus, and the Schmallenberg virus. Their vectors are midges, and their reservoir are birds. For the etiology of the disease, they are an SS single-strand RNA virus. They have a 3-genome segment. They're enveloped, and they have weak resistance, and they're arboviruses. They have cross reactions, but they do not have cross protection, and they're good antigens. For the epidemiology, the vectors are culicoidus species. They're midges. They're biological vectors, and they allow transovarial transmission. The host for Epi1 is cattle, sheep, and goats. For Onyo, it's cattle and sheep. And for Schmallenberg, it's also cattle and sheep. These viruses cause abortion and fetal development problems in pregnant animals. The reservoir are birds. They have a high amount of virus in the body, and they have a long viremia. The birds can play a role in spreading the virus over a long distance. The virus is urixin. Clinical signs are found in cattle, sheep, and goats, but susceptible animals can also be camels, horses, wild ruminants, and African swine. For the pathogenesis of the disease, a midge bite inoculates the virus into the body, and then there's a 1 or 2 day incubation period, and then viremia occurs. And this causes a problem in pregnant animals as it crosses the placenta and infects the fetus. And this can cause, number 1, abortion, or number 2, fetal development problems. In the clinical signs, so in non-pregnant animals, they're asymptomatic, or they can have mild clinical signs, such as fever, anorexia, and decreased amount of milk. And if it's a very virulent strain, you can see encephalomyelitis in the parent. In pregnant animals, there can be abortion at any stage in the pregnancy and fetal development problems, such as arthrogryposis, hydroencephaly, and muscle dystrophy. In the pathology of the fetus, we can see developmental problems, such as cerebellar hypoplasia, rubber limbs, so they bend in different directions, and longer or shorter bones. For the diagnosis, we look at the epidemiology, clinical signs, post-mortem lesions. In abortion, always remember to do the lab diagnostics, as there's so many different notifiable diseases, such as blue tongue or bovine viral diarrhea. To detect the virus directly, do a real-time PCR, isolation, or IF. And to detect the virus indirectly, do ELISA. For prevention, you need to have vector control, so you need to control the number of mosquitoes. And for abortion, close the herd and have movement restrictions and stamping out. And in endemic areas, there is an inactivated vaccine. The Schmallenberg virus is a new virus found in 2011 in Germany and the Netherlands. In cattle, it showed the general clinical signs of the OrthoBunyaViruses. So it's similar to Onyo and Epi1. The virus has spread from the Netherlands and Germany to worldwide, and it's spread by seasonal culicoiduses. So the occurrence is Europe and all over the world basically, and the pathogenesis, so the midge, which is seasonal in Europe, inoculates the virus into the body. And then there's an incubation period and a viremia for 4 to 5 days, and then it spreads. Non-pregnant animals are asymptomatic. Pregnant animals that are seropositive have no abortion because the animal has maternal protection. However, there's a problem in animals that are pregnant and seronegative. In sheep, if they're infected from days 30 to 50 of their pregnancy, they can have abortion and congenital disorders. In cattle, if it's less than 3 months of pregnancy, the brain and spinal cord is affected. If it's less than 100 days of pregnancy, there's joint and muscle defects. And if it's less than 6 months of pregnancy, they're immunocompromised. For the clinical signs, it's worse in sheep than cattle. But in cattle, you can see fever, depression, anorexia, poor condition, and decreased milk production, and congenital defects. They're rare though. In sheep, you can see a high fever, so around 40.5 degrees, and congenital disorders, such as arthrogryposis, which is joint stiffness, hydroencephaly, which is fluid in the brain, torticollis, which is a twisted neck, and scoliosis, which is a curved spine, and brachygynesia, and cerebellohypoplasia. And the lambs are usually born at the wrong date, and there's no breed discrimination. It affects all breeds. For the control measures, the disease has zoonotic potential, but it's not known. More research needs to be done, but it's unlikely, as no human cases have been reported yet. The disease is notifiable according to different countries. Russia and Morocco do not allow imports or breeding of Dutch heifers. And the USA, Japan, and Russia do not accept semen from bulls or embryos from the Netherlands. And there is an inactive vaccine.